Hello, welcome everyone to Learn Foodomics, Periodic Table of Food Initiative Science Trainings. We're excited for you to be here today. We're excited to share more about these trainings and to talk with you about everything the periodic table is doing to support Foodomics education. So I want to start our webinar today by introducing our panelists who we'll hear from later. First, Melanie Odenkirk. Melanie, would you mind coming on camera and saying a quick hello?
Hello, everyone. It's a pleasure to be joining you today. My name is Dr. Melanie Odenkirk, and I am a research scientist at Colorado State University in the United States. Happy to be here.
And Natalia Vázquez Manjarrez, would you also please say a quick hello?
Yes, of course. Good morning, everybody. I'm Natalia Vázquez from the Center of Excellence in Mexico at the National Institute of Medical Sciences and Nutrition, Salvador Subirán, where I am the PI of the Center of Excellence.
All right, let's get started. I want to begin by just acknowledging that for these periodic table of food initiative science trainings, which we'll get into today, this is really an exploration of the PTFI's research principles and standardized methodologies used for generating comprehensive food composition data. However, as we'll see today, this goes far beyond the PTFI and is available for anyone interested in learning more about foodomics in the lab and in the classroom. So Food EDU is hosting this webinar today. We are an open access e-learning and fellowships, or a platform that also runs fellowships. Our Good Food Fellows are in 36 countries around the world, 37 fellows doing work related to foodomics and the periodic table of food initiative. We're focused primarily on translational research and education that cuts across disciplines and has an applied component to it at the intersection of food, agriculture, health, and nutrition. Last chance for the poll. I'm going to close that momentarily, so get your questions in there, or get your answers in there if you haven't already. Okay. And I'll go ahead and share these results just to see, let you all see kind of where we're at with these questions. Learning more about foodomics is fantastic. That's what we're here for. And PTFI methods for those who are interested in teaching. Welcome. There's going to be some great resources here for you too. And it looks like we're pretty spread in terms of our overall familiarity. So it's nice, and we can be able to maybe learn from each other as we get into our discussion. So back to Food EDU. Again, we are this open access e-learning platform. Our curricula spans both disciplines and geographies. Our goal is to really promote scientific literacy, interdisciplinary collaboration, and to empower learners across the globe to address critical food and health related challenges. The Periodic Table Food Initiative is doing nothing less than mapping the world's edible biodiversity. Here we see a map of some of our centers of excellence and partner labs all around the world who are answering in part these questions. What's in the food that we eat? How does food quality, measured in this very comprehensive way, vary based on climate change, geography, production and processing in the agricultural space? And how does food quality impact human health and nutrition? These are some of the big questions that PTFI is grappling with as it introduces standardized omics tools to labs around the world to, again, map the world's edible biodiversity in ways that we'll learn more about in our webinar today. The PTFI science trainings, then, are a new series of trainings being launched by Food EDU on the American Heart Association's Professional Education Hub. Food EDU is a part of the American Heart Association, and that's where our courses are based there and have been developed, however, in collaboration, in close collaboration with folks from the PTFI, which we'll hear from today. These are open access, interactive, asynchronous e-learning courses. The modules are focused on one hand, which you can see here in the blue, on these very foundational scientific research principles. Principles of research design, data governance, stewardship, ethics, and food sampling, which we'll see a little bit more of today in my presentation. We also have a series of omics tutorials which dive deeper into the methods and the protocols to give you a really great foundational understanding of how to do this work in your own lab or in conjunction with others who are collecting these kinds of data. In addition to all of that, we have supplemental resources, including a learner guide, note-taking templates, and an online community forum, which you'll hear more about in one of our presentations today. All of which to support learners going through this journey of understanding better foodomics and these specifically PTFI methods and protocols. So who are these trainings for, and how might they be used? Really, the trainings are for students, researchers, healthcare practitioners, and educators, and generating and applying multi-omics data on food. We see this as being extremely relevant for labs interested in exploring foodomics and who might want to participate in this expanding global community of practice that PTFI is stewarding. PTFI is also contracted with national and partner labs and our quote-unquote centers of excellence around the world that have really helped kick off this initiative. And for those as well, these trainings are a fantastic resource to kind of go back to the beginnings a little bit. And a lot of these labs who have already onboarded different omics platforms might still benefit from understanding, again, sort of how these are situated in the broader context of foodomics and what some of those key scientific principles are going into this initiative. We see these trainings as also being extremely relevant to courses that are teaching mass spectrometry, courses in food science, analytical biochemistry, anywhere there are lab-based experimentation and you want to understand how you might use these protocols to teach something related to foodomics or food science or analytical chemistry. And we can have more of that discussion later. I'll be curious to hear from folks who are in this webinar, how they think they might want to use these courses. And lastly, for those who are interested in participating in the PTFI, who haven't yet entered into some kind of contract or are not yet a partner lab or a center of excellence, we also see these trainings as being extremely relevant to provide some of that foundational knowledge in a pre-boarding phase to what we usually refer to in the PTFI as onboarding, once you've contracted with PTFI and are onboarding the methods. So these trainings serve as a kind of pre-boarding to really get you oriented to what PTFI and what these omics methods are all about. So, what is foodomics? Many of you are here to learn what is foodomics. I am not here to teach you, but I do want to highlight one of the voices from a course that we've also developed at Food EDU called Foodomics and Society. And here you'll just hear briefly from one of our lesson experts talking about what is foodomics. This is Alejandro Cifuentes from Spain, who is often cited as having coined the term.
Hello, I'm Alejandro Cifuentes. I lead the Foodomics laboratory at the National Research Council in Madrid.
Pardon me.
It is a real pleasure to present the topic of foodomics and discuss its application in food science and nutrition. Omics sciences share the global aim of identifying, describing, and quantifying the biomolecules and molecular processes that contribute to the form and function of cells and tissues. As applied to food, foodomics is a discipline that studies the food and nutrition domains through the application and integration of advanced omics technologies to improve consumers' well-being, health, and knowledge.
There you have it, a wonderful overview from Alejandro from our course, Foodomics and Society. You noticed the timestamp perhaps on the top of that video. That's because this course is just about to be released in its entirety. We'll have 7 modules in this Foodomics and Society course that provide an even broader foundation to the field of foodomics and its applications in human and planetary health. The first 3 of these modules are available now on the Food EDU portfolio in the Professional Education Hub. Perhaps you'll see a link in the chat if you might, Britt. And we're really excited about these to engage a whole host of individuals, not just from the worlds of analytical chemistry, perhaps, and food science, but others in social science fields, in sociology, my background, and in other places that might want to understand better how food composition connects to these broader efforts to improve food systems globally and the health of humans and the planet. So with that, I am now going to do a quick preview and walkthrough of the Periodic Table of Food Initiative science trainings. And each of the 6 modules we have available now, there is a 7th coming still on glycomics, but we'll start with these first 3 that provide this foundational knowledge. In scientific principles and research design, and you can see all of these slides are taken directly from the course. There is a pretty broad focus here at the PTFI in that we are starting with these frameworks around sustainable food systems, the principles around global coordination and transdisciplinarity and community-engaged research are all foundational to how the PTFI has approached this effort. And as such, these are what we start with the course. And you can see from this graphic here, a pretty wide array of topics that we'll cover in this particular module, including standardization and data quality, which is also essential to PTFI and something we'll talk more about. In each of the modules, you can see things like this. These are knowledge checks that we have going throughout each of the trainings to really help test and assess knowledge as we go, and for yourself as a learner, perhaps to reflect on your own ability to kind of process information throughout the course. In data governance, stewardship, and ethics, we go more specifically into topics such as access and benefit sharing, data stewardship, the FAIR data principles, ethics, equity, data sovereignty, indigenous data sovereignty. This list here is just to show you an example of how we begin each module, showing you an outline of where you're going to be going, and also describing a little bit more about the rationale for how we got there. In this sense, things like access and benefit sharing are also essential to PTFIs. We think about collecting potentially sensitive data from crops that have been stewarded by, for example, indigenous people for millennia. It's really important to understand how we can best share and honor the responsibility that comes with this work. So this is something that's super foundational to PTFI, and also, again, why we put it in this series that's really meant to provide that orientation to the effort. In food sampling, which might seem a little bit more straightforward, it can be actually quite complex. How we collect these foods, how we process them in order to generate these data, can come with things like metadata, which is also important for the PTFI as we put these foods into context, not just where they were grown, but how they were grown, how they were processed, all of this having an effect on how we understand these omics data that are being generated. There's preparation decision trees in this module, for example, that can show you how you might go about preparing things like fruits and vegetables or leafy greens, something like this. And this is meant to provide a hands-on guide to anyone who's actually processing foods and collecting those metadata to really ensure that the data that's generated and assembled into our database is consistent, standardized, and reliable. We move from here into our omics tutorials. I'm going to go relatively quick through this so we can get to a video I want to show you at the end. Our ionomics tutorial, again, begins with some fundamentals that might be more familiar to those of you who have studied ionomics, and then goes more specifically into some of the methodologies that are related to PTFI, and with beautiful illustrations such as this. Similarly, in lipidomics, we're getting into, again, some of the more technical details about what this particular protocol entails. But also its research applications. What are some of the ways in which these data can be applied for those broader benefits that we want to see mentioned in our foundational principles? To talk more about metabolomics, I'm actually going to let all the speaking go to one of our subject matter experts who will be coming up momentarily. This is a preview, and I'm excited to share this video with you. Each of our tutorials also has short video components such as this.
The diversity of biomolecules in the food we eat is vast, including essential molecules like fat, carbohydrates, and proteins, as well as a wide array of small molecules called secondary metabolites. These compounds are made by plants in response to stresses such as disease, climate, and predators. But there is also evidence that these molecules are good for us as humans when we consume the plant as food. The biomolecules in food come in all sorts of shapes and sizes, and this diversity means that we cannot measure all of them with the same analytical method. So within the periodic table of food, or PTFI, we've worked really hard to look at all of these small molecules in a very regimented way. This is important because while we have the technology to look at small molecules, typically the labs that are doing this all do it differently. So their answer to questions like what's in an apple, what's in a tomato, are not the same, not comparable. So much chemical diversity in the small molecules of food, we use two methods: a reverse phase separation to resolve nonpolar molecules, and a complementary hydrophobic liquid chromatography, or HILIC column, to see the polar small molecules in our food. As the molecules elute from this column, they are introduced into the mass spectrometer, where a high voltage is applied, resulting in the generation of gas phase ionized molecules. These charged gas phase ions are then directed to a mass analyzer that separates analytes by their mass to charge, or M/Z value, prior to detection. This data acquisition can be visualized in real time by a chromatogram, which shows us the signal intensity for all of the detected molecules over time. The resulting data files are uploaded for further processing using the standardized PTFI data processing workflow, enabling evaluation of relative intensity changes for molecules that match to a global consensus library of formula and authenticated standards. With this method, our team at the PTFI is working to understand how these molecules serve the plant in defense against external stressors, such as climate change, and how these molecules translate to color, taste, and health benefits when they reach our eyes, mouth, and stomachs.
That doesn't make you excited to enter the fray. I don't know what will. And one more thing I'll say, these are examples of some of the learner guides and the note-taking templates associated with each of those modules for our trainings. We think they're really excellent resources, again, to support you and your learners who are coming along on this journey. Sign up now. You can access the trainings. There's a link in the chat, or we're on the Food EDU portfolio on the Professional Education Hub. So thanks everyone for your attention. And with that, I'm going to stop sharing my screen and turn it over to Melanie Odenkirk to talk a little bit more about how she came into this PTFI journey and what these trainings could mean for you and yours.
Hello, everyone. It's wonderful to be here today. My name is Melanie Odenkirk, and I am a research scientist with Colorado State University. Today, I will be talking about breaking bread and barriers with Food EDU science trainings. To tell you a little bit about me and my background, I have been doing analytical chemistry research for a decade now, and this really started in 2015 with my first experiences with mass spectrometry in a lab in Virginia in the United States. Here, we were looking at an ionization technique that's called electrospray ionization. And this was my first experience with mass spectrometry, which is a powerful analytical technique, not in that it allows us to measure a wide array of molecules, but it also provides mass information about the molecules that are present in a system, which can be incredibly informative when we're trying to understand what is part of a system and what might be changing as related to specific stressors. With that, that foundational work with mass spectrometry brought me to a PhD in analytical chemistry at North Carolina State University. Here, I took some of those fundamental techniques that I had originally learned, and we started applying them through omic-based analyses, where we're measuring specific subsets of molecules. And in this research group, we were primarily looking at biological studies and trying to understand how these molecules were important and how they would change through a variety of applications. Specifically, these included a series of singular omic applications, such as looking at changes in small molecules before and after exercise and myocardial infarction events. Also looking at stem cell differentiation into neural and mesodermal lineages. I also did a series of multi-omic studies where we would take several analytical workflows optimized for specific types of molecules and combine the data that we would get, such as lipid and protein data, to understand disease pathophysiology of things like preeclampsia, gestational diabetes, and Alzheimer's disease. This work brought me to a postdoc in analytical chemistry, where I was again looking at the interaction of molecules, specifically membrane proteins and their surrounding lipid environment. And in this work, we were primarily looking at E. coli and brain-based systems. From here, I made the transition to the role I am currently at with Colorado State University, from biological samples all the way into food. And food is both biologically and chemically very diverse, much more than what we might see in a biological-based sample. And so the aspect of standardization is incredibly important. And so our ongoing collaboration with the Periodic Table of Food Initiative, or PTFI, is really important as I have made this transition into working with food. Namely, to give you an idea of what I mean when we're talking about standardization, it's really the end-to-end workflow of actually going out and acquiring food all the way to generating our final data set. So this would begin with the process of food collection, which we have standardized and best practice guides, whether this be in your local garden, from a field, or from a market, the documentation of metadata, and food preparation when it gets to the lab. For metabolomics analysis, we also have a standardized practice of doing small molecule extraction with solid phase extraction and standardized reagents for this as well. For the actual data acquisition, we also have a standardized method that includes a reverse phase LC separation with a standardized gradient, collection of data that's MS1 with high resolution instrumentation, and optional collection of both DDA and ion mobility data. Finally, we also have a standardized data processing pipeline developed by Verso Biosciences that allows for the alignment of this type of data, not just across different laboratories, but also different instrumentation, and a curated final data return of all of this, this work that's been done. And to give you a little bit of an idea of how I came to find really how important this standardization is, I want to tell you a little bit about my first analysis I did working with food. And here I'm showing you a chromatogram of a carrot sample that we ran in a project where we were looking at carrots and apples. What you see here is retention time on the x-axis and intensity on the y-axis. And all of the lines here represent different molecules that we're seeing as we analyze this carrot. And I injected this carrot and then shortly after injected this apple sample, which you can see even with all of the standardization we're doing, the actual peaks in the molecules that we're seeing across these foods are so different. And even though I had been doing mass spectrometry for a really long time and applying this to a variety of different studies and sample types, this chemical diversity was so great that when I first saw this data, I thought something had broke in the instrument, because it was just such great chemical diversity, more than I had ever seen in biological samples. But this is just the reality of food. And this is a huge analytical challenge because not only is this chemical space very different, the biological diversity of the food on our planet is also vast. So take, for example, a banana. This is an example of a banana we commonly get imported and eat in the United States, but this is actually one of 1,000 varieties of bananas. And across all of these different banana varieties, we might have changes in nutrient density, resilience to environmental stressors, and flavor profile changes. And on our planet, the edible biodiversity is over 23,000 species. So this is obviously a huge analytical challenge. But when we think about trying to measure all of this space and what we're really trying to measure it for, which is to improve our food system and address challenges with human and planetary health that food can directly support, this is really something that bridges just analytical chemistry to be a true transdisciplinary challenge, where it's not just an analytical chemist, but a team of researchers and people around the world who have unique insights and backgrounds that are all contributing to our food system in some way and can help make it better. But there's a real challenge with actually integrating all of these different disciplines together. There are barriers that exist across all disciplines that really kind of hinder this transdisciplinary need for this application. Specifically for analytical chemistry, there are barriers of technical terminology, constantly changing best methods of analyses, and also really large, complex final data sets to work with to really understand what's going on in that system. And this creates a very figurative, but that can feel very real barrier to actually applying analytical chemistry, which is very powerful to understand our food system and how to better it. And so really our goal with developing these Food EDU science trainings was to start breaking down these barriers. Namely, what we aim to address in all of these science trainings is why these molecules are important in food, how these molecules are measured and how that might impact our data, and how to correctly interpret final data sets. I had a role in developing this for both the lipidomics and metabolomics modules, but as Kevin mentioned, there are other expertise in areas that are also covered through the Food EDU science trainings. And this has been really beneficial to me as I have transitioned from doing biological-based samples to working with food. Through this food sampling module taught by Dr. Sarah Brinkley and Margaret Reed, I've come to learn about unique sampling strategies and handling procedures for working with food and really applying best practices. Additionally, I've learned a lot about ionomics, which is really important for understanding the macro and micronutrition of foods as they get to people and making sure that they're getting the nutrition that they need, and how to actually implement this for a wide variety of food matrices. Further, I am involved in co-creating a course that will be taught at Colorado State University at the graduate level for spring 2026 called Mass Spectrometry Omics. And we plan to integrate some of these modules, such as the PTFI metabolomics modules, to build out some of this foundational knowledge and how this technology works, and specifically how it can be used for food, which, as we've mentioned, has much more complicated challenges for really understanding that chemical diversity and coming up with a method that will be suitable for working with different food matrices. While all of this training effort is obviously really important to starting to break down these barriers, we also need to start thinking about how we can facilitate some of these connections. And so in addition to all of this work with the Food EDU science trainings, we also have an online ecosystem that has a QR code here and will also be shared in the chat momentarily, a link to join, that allows a community of scientists to come together to connect that are all looking at food systems, to explore some complementary resources that support food research, and to also collaborate to tackle some of these complex questions at the intersection of science, food, and society. And so ultimately, our vision through Food EDU science trainings, the development of this ecosystem, PTFI standardized methods, is to bring everyone to the same table, because we recognize through all of these disciplines that touch food, everyone has something to bring to this table and something to learn through some of these resources. So I'll leave you with this last thought of joining the table and thinking about what you might want to bring to this world. I'd just like to thank all of the Food EDU content creators, everyone who supported the development of these modules, the PTFI team and all of our collaborating labs, and the laboratory at Colorado State University. Thank you all for your time.
Thank you, Melanie. A wonderful presentation, really insightful as we think about what it means to be doing this work in relation to food in this broader field of analytical chemistry. And I'm excited to have more of a discussion about that. after we hear from our next presenter, Natalia Vázquez. Natalia, I'll turn it over to you.
Thank you, Kevin. I'm sharing my screen now. All right. So hello, everyone. Again, I'm very excited to be here today and talk to you about my journey in Foodomics and the path we follow to build a local capacity where PTFI and Food EDU have been crucial. I was trained as a clinical nutritionist. And from the beginnings of my career, I've always been trying to understand which is the link between food and human health, not only from a macronutrient perspective or a caloric perspective, but also on a molecular level. I started working with regional foods such as nopal and understanding its impact in the incretin effect in type 2 diabetes. I then moved forward into studying a collection of different foods, of functional foods that we call a dietary portfolio, and how this impacts hypercholesterolemia, specifically on the lipoprotein profiling that's measured by NMR. By understanding everything and the good that foods can actually do to human health, pushed me into pursuing a PhD in nutritional metabolomics, where I was fortunate enough to be taught by Lars Draster and Claudine Manac under the umbrella of the Football Initiative. Football Initiative had the aim of discovering biomarkers of intake of the most consumed foods in Europe. And it is there where I learned how to work with untargeted metabolomics to do data analysis and understand all these different patterns that the data was telling us. Later on, I moved on to Mexico again. And there I lead the metabolomics unit at the National Institute of Nutrition. It is my aim and our aim to approach or to use omics approaches to understand nutrition and bring them into public health. But throughout my journey, I was able to see different problems and obstacles into applying omics into our fields. We could see in the application of nutrition that we still had bias in dietary records. With this bias in dietary records, we would have information that is not robust enough to build association studies with health and diet. On the other hand, the food composition still remains a black box. And this is a food composition that we are using to build dietary recommendations and design our dietary therapies. Therefore, it came as no surprise that dietary recommendations built in our country today are mostly ineffective. I also became acutely aware of the communication barrier or obstacle between the people that do omics scientists or the omics science as well as other disciplines. At heart of these communication barriers was a lack of training and knowledge of what Foodomics is able to do. But this is exactly the kinds of gaps that Food EDU and PTFI are trying to bridge, not just by generating better data, but by building a global interdisciplinary community and developing shared standards and trained sources. To understand the value of PTFI and Food EDU, I often like to think about the Rosetta Stone. Discovered in 1799, this Rosetta Stone contained the same degree in 3 different languages: Egyptian hieroglyphs, Demotic, and Ancient Greek. But it was not until the 1820s, thanks to Jean-François Champollion's ability to speak and understand Ancient Greek, that he was able to translate all these different Egyptian hieroglyphs in full. With that came the modern understanding of Egyptian language, and with it, a deeper insight into Egyptian culture. Talk about a cross-cultural breakthrough. Just like the challenge with Egyptian hieroglyphs, Foodomics faces a similar need to translate the complexity of the food composition obtained by these different omic technologies and put them into biological meaning. This requires interdisciplinary tools, a shared scientific language, and critically, more people who can understand and apply omics technologies. That's exactly what the PTFI and Food EDU platforms offer. Through targeted training, Food EDU is laying the groundwork to build bridges between disciplines and enabling data-driven solutions across agriculture, health, and food systems. By achieving this, we may be able to create a cross-disciplinary breakthrough for Foodomics application. In our Center of Excellence, we are launching 2 different demonstration projects. In hand with PTFI and Food EDU, BMX and MAPAMX have at heart the use of untargeted metabolomics and lipidomics methods. For BMX, we aimed at the discovery of biomarkers of intake of Mexican foods, where we hope to obtain molecular markers that we are able to trace back to the food itself in order to be able to have a better dietary accuracy in association studies. On the other hand, with lipidomics, we want to obtain further profiles aside of the fatty acids in order to understand how is it that these intact lipid species play functional roles both in foods and in human samples. However, we had the instrumentation for lipidomics but not the know-how, while for the untargeted metabolomics we had the know-how on how to apply the data but not on the instrumentation. Here is where PTFI and Food EDU came in. For the lipidomics method, we worked very closely with Kevin Williams at UCLA Lipidomics Core, who is a method developer, and in hand with Melanie Odenkirk at the Core Lab, who is our network lipidomics expert. We knew that onboarding wouldn't be easy, and it of course took a lot of effort, patience, and time. As it is often the case, as you can see from this picture, we encountered a lot of problems. At some point, we couldn't really get any stabilized signal throughout the run. Ionization looked completely off, as you can see. But after doing some troubleshooting and communicating constantly with Melanie and Kevin, we were able to pinpoint the problem, and we realized that the electrode became dislodged from the source when we tightened the LC line in tightly into the IS. As you can imagine, this is just one of the many challenges that we face that came from flow, things that came from ionization inconsistencies, and many others. But what helped us move forward was having a shared language between Kevin, Melanie, and our Center of Excellence, as well as for our Good Food Fellows. This is where food training really helped us to get this communication going. In the lipidomics training, as well as for the other omics trainings, you can find the who, the how, the what, and the where of the different methods. In the specific case of lipidomics, we were able to understand the who are we going to measure, so the different classes, categories, species, what that actually means, the difference in structure of the different lipids. On the how, we were able to understand what DMS is, how it works, and what is needed to obtain successful acquisitions. We also were trained on the where, meaning which is the software that is needed in order to make all the raw data process, and then be able to obtain the what, the data sets and the data matrices that we are going to mine in order to obtain results and answer our research questions. So at the final result of our onboarding, is that we were able to obtain good acquisitions and final pre-processing of the raw data using SLA software, allowing us to now start to be able to answer the questions regarding the foods in relation to nutrition. So we then started questioning all acquaintances in our foods. We started looking at the raw data and we started looking at the raw foods of chicken, egg, beef, and fish. By looking at this graph, we observed how principal component analysis clearly separates all these different foods according to the lipid profiles. As a nutritionist, that really impressed me. As you can imagine, usually in some countries, such as mine, when we're talking about animal food origins, we often use the term interchangeable between each other. Based on this result, we can actually see that that is not true, as they are very different from each other. We went even deeper, and then we tried to see which was the difference between all these different foods. As you can see from this purple clad, which stands for the triglyceride species, they are very apart from each other, pulling what seems to be fish away from the rest of the foods. When we then highlighted these different triglyceride species according to their level of unsaturation, we could see that those triglycerides in fish have a higher number of double bonds. As we continue working on the data and collecting information of other foods, we then we then encounter another obstacle. We wanted to go a little bit deeper into bioinformatics and understanding better what is it that is in this data. For that, we then again went back to our partners in the core lab, where they taught us how to use specific bioinformatic tools for lipidomics, including SCOPE and LipidR. We're now able to produce richer results and statistics that allow us deeper comparison between foods, agricultural practices, cooking methods, and much more. Thinking with my dietitian hat on, I can already imagine a future where dietary designs is based on data-driven approaches using food composition data. But what now about our own targeted metabolomics data? Well, again, thanks to the collaboration with the core lab, to date, we have obtained 75 putative urinary biomarkers that are specific for the intake of tortilla, nopal, mango, guava, and avocado. As you can see from this heat map, those that are in a very high yellow, it means that they are in more abundance in those foods after their intake. Well, we know that there will be much more work to come regarding identification of these features, as well as a validation study to be able to understand their robustness. This lays the groundwork for us to be able to obtain a better dietary accuracy for association studies. From our work, I can tell that Food EDU and PTFI together have the power to lay the foundation for sustainable foodomics. By training the next generation of scientists, sharing tools, establishing a common language, and fostering a global interdisciplinary community. So, get on board. I would like finally to thank everybody who has funded our work, as well as the PTFI initiative and Food EDU, and all the people within our Center of Excellence who has helped in developing these projects. Thank you very much, and I'll be happy to take your questions.
Thank you so much, Natalia. Wonderful, wonderful presentation. I especially love the idea that this Rosetta Stone of food composition is what PTFI is bringing to the table. I'll invite Melanie to join us, and you can stop sharing your screen anytime, Natalia. And we do had a couple of questions coming in through the Q&A. I encourage you all to continue putting your questions in there. We'll monitor them as they come. One question that had just come in was about this kind of training pipeline in a way. And so I wanted to talk about that briefly, just to say that these courses that we're offering, the Food EDU trainings, including the Foodomics and Society course, are completely free, open access at any time to anyone. And in that way, those are asynchronous, meaning you do them on your own time and with whomever you would like. We do have supplemental resources to go along with those trainings. But Melanie, I wonder if you can talk about the process for what it looks like for labs that are working a little bit more closely with PTFI, perhaps that are contracted labs or national partner labs, and what that means in relation to your team and other onboarding activities.
Absolutely. And so we are offering several tracks for getting involved in PTFI. We have labs who are interested in applying these methods that maybe don't want to use the full sweep of PTFI. That is also an option with the standardized framework that we're developing. While we have shown it for food, it can be applied for a lot of various samples, such as biological studies as well. And there are definitely pathways for that. But for being involved in PTFI as a lab that's doing the full workflow, we have, in addition to some of these pre-boarding activities, onboarding experiments and opportunities to go through and actually do the food sampling, applying the analytical method and getting validated to ensure that the data is of high quality and is meeting our standardized criteria, so that when you go to do your research projects, you can have the utmost confidence in that result. We also have a PTFI database that right now houses 500 foods, and we're continuously working on growing. And this is also a place where labs that are working intimately with PTFI, their end data will ultimately go, so that we can better understand all of the food that's been measured in all of these different analytical assays and combining that together. I encourage you to explore that resource as well.
Thank you, Melanie. Natalia, you've been through this process largely in multiple platforms. Can you talk a little bit about what that onboarding looked like from your experience there in Mexico?
Yeah, our onboarding was amazing. We had an in-person training with Melanie for lipidomics where we had a lot of fun, obviously, and she prepared us to start doing this analysis afterwards by our own. Like it always happens, when she was there, things ran very smoothly, and then she left, and we started having all these different and weird things in the machine, as I have shown today. But it was amazing. It was very good to have a constant contact with her. This is something that I do constantly with email, constantly through Phyto Hub, through the hub that we have that Melanie has mentioned today, where we can have conversations about what can we do, the problems that we are facing. And what feels very nice is the fact that we can have a check in with Melanie in terms of the quality of our data. So we do know that we are confident of the results that we are having, even though this is a new technology that we are addressing.
Thank you. Yeah, it does seem like, and based on my experience in working with centers of excellence in this project so far, that relationship has been essential, right? So I want to just highlight that in terms of the onboarding process overall, and I'm putting a link in the chat, you can express interest by looking at the PTFI website, and there's a link at the top to join the PTFI and express interest in adding your lab to the list of those that are onboarding these methods. That the interactions with the team at Colorado State University and the Prenni lab have been instrumental, it seems, for all of our centers of excellence and all those who are joining PTFI. And we are hoping that these trainings and others that Melanie alluded to from the PTFI Ecosystem Hub can really just accelerate that effort, and provide some more of these asynchronous resources, so that Melanie can continue to do other things. Not that you don't always love talking with folks like Natalia. Speaking of which, you both came from different backgrounds, analytical chemistry, clinical nutrition. What was it like to have some of those early conversations, knowing that you did have those disciplinary distinctions? Were there language barriers to overcome? What was some of the things that, and I know, Natalia, you had gone into the omics through some of your own clinical work, but what were some of those early interactions look like, knowing that you had come from these different disciplinary backgrounds?
The communication, as I said before, was essential. I think what really helped us was to have different perspectives on what the data should look like and the discussions that we have regarding the data. That has been very nourishing for my perspective. I hope for Melanie as well. And that really helped a lot into onboarding because as we have the same language, not 100%, but since we have a good communication with each other, meaning that I understand when she's talking to me about specific terms, and the other way around, I think it's facilitated a lot of the onboarding and accelerating the results that we're having.
Mm-hmm. Yeah.
If I can add to that, Kevin, yeah. I would just completely agree. And I would say also in our, like I support Natalia's onboarding activities and her lab independently running lipidomics, but we also collaborate on metabolomics projects. And that is really wonderful because the way I think about a problem is different than Natalia thinks about it. And so we go about tackling it in different ways. And my way is not always the best way. And so Natalia's different perspective is really valuable, especially when we get back these final data sets, you know, that biological understanding of what could this mean. Natalia has all of that knowledge. And so it's wonderful to be working with her because she has such a rich resource of knowledge. And I think that that's true in a lot of these collaborations as we bring people together. By creating that safe communication space, we're making each other do better science as well.
I love that. You know, and there was a question in this in the chat in relation to different varietals and standardization, you know, from within a sample, right? Can we see different Gala apples look differently based on perhaps where and how they were grown? And this, I think, speaks to some of the metadata kind of contributions and challenges associated with this project, and ultimately where the PTFI might have the greatest impact and be able to situate foods within these contexts, right? And what that means for food quality overall. So Melanie, maybe we could start with you. Like, what have been some of those kind of breakthroughs? I mean, you talked about how food is different in this space. It's more biologically diverse, but the metadata that's being collected along with these foods in this project might also be different. So can you talk a little bit about maybe what that's meant for you and what some of the challenges might have been to kind of integrate the metadata?
Absolutely. And I would completely agree. I think that this is a fantastic question. And to me, this is what we're aiming to address with PTFI, because not only do we have all of these, biologically diverse species of food that we're eating every day, but also all of these factors that are impacting them, it's an impossible space to think about measuring. And that's why standardization is so important. And that really starts with cataloging how the food was grown and getting all of that information. So as we're generating this data into databases like PTFI, we'll eventually create a big enough network where we can start teasing apart how these parameters might be influencing some of this data. And I don't think that really to understand all of this in food, this is anything one lab can address on their own. And so it's been a really kind of eye-opening thing that even with all of my expertise, we really need to all rely on each other. And that comes with coming to the table and being willing to change maybe how we would typically do things so that we can get things that are comparable to really start answering those bigger questions.
Mm-hmm. And different kinds of questions, right? Maybe that haven't been fully articulated in the past because these methods didn't exist in this way. And Natalia, you're looking also at the sort of clinical and nutrition side of things and introducing biomarkers to the equation. So on one hand, we have metadata that might capture maybe its provenance and how it was grown. What for you does it mean to kind of then take that and also be looking at biomarkers for the human body and then combining those massive data sets?
I love that question. It's a very good question. I think what you described is exactly what we want to bring into precision nutrition. Or precision medicine as well, to be able to do the data fusion between what we find in the biological matrix regarding, for example, a specific disease like the ones that I've talked today, like diabetes or perhaps hypercholesterolemia. In our case, that is very important because we are we have a lot of metabolic diseases in our country, particularly those two. So it would be amazing to be able to understand how is it that the biomarkers for those diseases work in our population as well as in others, and how that works with food. By that, and joining those two and applying artificial intelligence and AI, we may be able to get closer into making dietary designs that are much personalized.
Wonderful. Yeah, the precision element, right, is a fascinating one in functional foods and precision medicine. Also something that the American Heart Association is involved with, and exciting to kind of be able to participate in that space. A question coming in from the chat. I'm just going to ask it directly because I feel like it has a relationship to all these things we're talking about. I'd like to ask if your work can help improve crop quality by eliminating non-beneficial nutrients and perhaps reducing anti-nutritional factors, ultimately making the crops healthier and safer for human consumption. And one of the reasons I like this is because it has to do with agronomy, right? And suddenly now we're talking about clinical nutrition and analytical chemistry, and now we're talking about agronomy. So Melanie, what do you think? I mean, what is the role that this work can play in helping improve crop quality?
I think this is a great question, and this also speaks to how multifaceted growing crops is and all of these factors, and comes back to metadata. It all always comes back to metadata and the cataloging of all of this data that these labs around the world are collecting in a database, so we can really start answering some of these bigger questions. I absolutely see this being a part of the future. But the foundation is building out these training resources, like building this knowledge, communicating together to start addressing these questions. And I think that this is exactly where we envision PTFI and resources like this enabling, researchers around the world to go.
Wonderful. We have another question coming in the chat around, will there be, with all this variety of data, will there be statistical analysis modules as part of the science trainings? And I'll just say briefly that one of the things that we're looking at towards the future is that data analysis component of this, recognizing that, just as you showed in your presentation, Natalia, that understanding some of these data that are coming out of the runs through the mass spec and such requires maybe a different set of tools as well, right? Or a new set of tools to be thinking about these data in the context in which they're being studied. So not quite yet. I think it's in the making, and excited to kind of think about that for our next phase. So thanks for that question, Amy. Any opportunities for young dietitians like myself in Ghana to collaborate or receive training on our local foods and omics research and lab technologies? mostly because we access nutritional content of foods using just the food composition templates. So I think with that, I might sort of give a plug for 1K in UST Ghana, Jocelyn. So if you're not already connected, right, we have a partner lab in KNUST Ghana and Kumasi, and I think that that would be one interesting place to start. Ideally, we're building out this network globally such that the local food composition data tables can become more robust, that data being generated on local foods is being conducted by folks in those places, in those environments, with the foods that are relevant to those communities. In this distributed way, we're able to expand the world of food composition and the world of food, in that we are able to collect more of the foods that are relevant to the communities worldwide and make that data meaningful to those people in those places. So with that, I want to do one last poll here before we wrap up. And let's see here. As I'm doing that, Natalia, and we'll start with you and then Melanie, any sort of closing words that you'd like us to consider?
I'm happy to go, and then I'll hand it over to Natalia. I see some questions in the chat, and I think that this really summarizes what this webinar is about, that we're really hoping to bring everyone to the table and start breaking down these barriers with Food EDU. And so these are resources that are open and freely available to anyone. So even if you're not actively onboarding a PTFI method or in that phase, you can still use these resources. Join our community. It's not just for onboarding, it's for connecting with people who want to better understand food for a variety of different things. So we encourage you to join these spaces as we kind of grow and populate them more. We're also really interested in what you feel like would be most beneficial for you and where you are and how we can be facilitating that. So please, give that feedback to our team in some of these spaces, and we're happy, to best support that however we can. And Natalia,
fantastic. Just really quick, Natalia, I don't want to jump in, but those things are exactly what we want you to do in the poll that's currently live. Suggest what topics of interest we should be focusing on for our next series of trainings. How can we create asynchronous or synchronous events like this that are meeting your needs? Put those topic areas in the poll. And Natalia, I'll turn it over to you for our last word.
Yeah, I would just like to encourage everyone to join the Food EDU and PTFI if possible network. In our experience, it's been more than enriching. We're now working so much closely with not only with the core lab, but also with other centers of excellence, such as UC Davis, for example, where they have infrastructure that we do not have, such as proteomics, for example. And we are now doing a lot of collaboration as well in order to push forward how is it that food composition can help us have a better planetary health. So don't doubt into diving in. It's a wonderful network to belong. And please come into the ride with us.
Thank you, Natalia. Thank you, Melanie. Thank you, Britt, in the background monitoring our chat. Thank you, everyone, for being here. We really appreciate it. There is a link to the science trainings in the chat. Click on it before you go. You can have it for as long as you like. It's open access and it's free. We encourage you all to join the table. And once again, thank you both for wonderful presentations. Thank you all for being here.